All right, so hey class, for our presentation, we're gonna be talking about human iPS cells derived from dopaminergic neurons and how they function in primates, Parkinson's disease model. So for our specific presentation, we'll be showing you guys figure 2A through C, what these images mean, and we'll go in depth a little bit later on. So the people presenting in this project is gonna be Erin, Lucy, Eric, and Jenna. So a little background on this information is,
Okay, so what is Parkinson's disease? Parkinson's disease is when when there's a loss of dopamine. So if you can see the image on the right, it shows a normal neuron transmitting dopamine. So all those little dots right there, that's dopamine being transmitted to the brain. And this person clearly does not have Parkinson's disease. So dopamine is very essential for motor skills. It's made in the part of the brain called the substantia nigra, which can be found in the floor plate. Dopamine making neurons are made from dopaminergic neuron progenitors. So when you have Parkinson's disease, you may experience the acronym TRAP, which stands for tremors, rigidness, akinesia, which is abnormal movement, and posture or balance issues. It isn't just affecting bodily movements, it also causes depression, anxiety, breathing problems, muscle weakness, and so much more. This disease affects men the most and females not quite as much. This research is very important because there is currently no cure for Parkinson's disease, but there is a very common treatment called L-DOPA, and what it essentially is a synthetic dopamine that is put into the brain to try and replace that dopamine that isn't getting there. So the only problem with L-DOPA is that there are side effects. So scientists and researchers are trying to find another solution where the side effects won't be a problem. The new solution is to use iPS cells, which are induced pluripotent stem cells, which mimic what embryonic cells would do if you were to use them in the body, but it isn't ethical to use embryonic cells. You first obtain the cells from the patients who have PD and you force them to become iPS cells. Those iPS cells are then made into progenitors that can potentially make dopaminergic neurons. And finally, they're transplanted into the PD primate models. And then long-term testing and experiments will take place.
So the portion of the study that we covered was meant to determine whether the transplanted dopaminergic neurons were effectively grafted into the brain. And the methods used were MRI scans and immunostaining. So once the dopaminergic neurons were transplanted into the monkey brains, MRIs were used to examine the areas of the brain in which the neurons were placed. Monkeys that received dopaminergic neurons derived from patients with PD, monkeys that received dopaminergic neurons from healthy individuals, as well as monkeys treated with L-DOPA, all underwent MRI scans on a monthly basis up to 24 months after the transplants. MRI stands for magnetic resonance imaging, and it uses strong magnetic fields and radio frequencies to obtain images of the body that cannot be seen well with X-rays and CT scans. The MRI works by applying a super strong magnetic field, which aligns the proton spins in our bodies. And when that magnetic field is shut off, the protons gradually return to their normal spin. This return process produces a radio signal that is measured by receivers in the scanner and made into an image. On the MRIs, the grafted areas were identified to be the darker areas of the scan. And these MRIs were used to estimate graft size from time of transplant up until euthanasia. At euthanasia, graft size were measured using a different form. We used STEM 121 staining, which is a type of immunostaining. Immunostaining is a method where an antibody is used to detect a specific protein, and fluorescent dyes are bound to the antibody in order to visualize the proteins being detected. In this case, the antibody used was STEM-121. STEM-121 is a mouse monoclonal antibody, which is specifically used to detect engraftment migration and differentiation of human cells after transplantation into animals.
So here we have figure 2A. Figure 2A is a T2 weighted image of the grafts in the monkey's brain. T2 weighted MRI scans help us see contrast and brightness in different tissues, which highlights the transplanted cells in the brain. The grafts are the bright areas that the arrows in the image are pointing to. This image was taken 11 to 12 months after the cells were transplanted. By looking at this image, scientists are able to analyze cell growth as well as survival, migration, and any abnormalities that might affect the safetiness of this treatment. Next slide. Here we have figure 2B, which shows the graft size over the 24-month period. It shows us the results from the vehicle group, healthy group, and PD group. We can see in all the groups that the growth of the graft cells plateaus around 6 to 9 months. Extended figure 3A shows the maximum growth of the graft sizes over the 24-month period. It is important to see how large these grafts will grow and see how safe this treatment is. Next slide. Figure 2C compares the graft sizes in the healthy monkeys to the monkeys with PD. This table shows us how the graft sizes differed in each monkey. The average size was 39.4 plus or minus 21.2 millimeters cubed. This is important because it shows us that this treatment won't cause a mass effect or a tumor. Figure 3B compares graft sizes from MRI imaging and the immunohistochemistry imaging, and we can still see that the graft sizes differ in the two different types of imaging, which kind of confirms that the graft size differed in each monkey.
So what are the results and conclusions? Starting with figure A, which can be seen on the top right, the hyperintensity images in the T2 weighted images possibly include an edema, cell migration, or neurite extension. An edema is a medical term for swelling, which usually occurs when small blood vesicles leak fluid, and in this case would be in the brain. It could also be cell migration, so cells are migrating to the origin of the brain where Parkinson's affects it. Or it could even be neurite extension, and neurites are beginning to grow, which would be a good sign. However, we're not exactly sure which one of them it is right now, but we do know that growth is occurring, which is a good sign. So for figure B, which can be seen on the bottom right corner, during an 18th-month period, graft cells showed an average size of 50.73 millimeters cubed. The majority showing an increase in graft cells throughout the first 6 to 9 months and then slowly plateauing after. But overall, majority of the monkeys were increasing their graft cells. The highest estimated of them was 106.77 millimeters cubed with a 95% confidence, and this was taken at 24 months prior to the injection. So for figure C, calculated graft volumes based on STEM-121 staining differed in each animal. What this means, it indicates that these grafts were unlikely to cause a mass effect. So for a mass effect, it could be contusions around the brain tissue, it could be edemas, cancer cells. Overall, it's something negative that you don't want to see inside the brain. So not having any of this is a good sign. This can also be backed up with the MRI values and histological analysis, which you can be seen in the middle image. Using the scatter plot, you plot each and every data point that you have with both of these tools that were used, the MRI and histological analysis. Using a line of best fit, which was y equals 0.74x plus 12, you were able to gain a R value. An R value basically measures how closely correlated each graph is to one another, or each plot is to one another. The majority of this, you want it to be closest to one as possible, which indicates that they are in fact closely related, and having a 0.92 is a good sign and a positive sign. Right below that, we also get a p-value. So for the p-value, unlike the R value, you want a small number. You want it as close as 0 to possible. The closer means that the stronger the evidence is. So having a super small p-value, and in this case we do, is a good sign showing that our evidence is strong. So what does this mean overall? What does this mean for research? And the grafted cells are healthy. Both MRI and histological evidence back this up. And most importantly, the cells are growing, they're surviving, and they're doing healthy inside the brain.
So we see that the results are good. The cells are surviving, they are healthy, but there's still some concerns about this research. We need to see if we would get the same results in humans as we did the primate models. We should have a bigger sample size because there were only 11 monkeys that were tested on. We need to add randomization, and what that means is randomly putting the monkeys in each group because there were none, and that could mean there was a sample bias. We need to understand why there was an unsuccessful result, because as we know, there was one monkey that did pass away. We can observe the models for a longer period of time before euthanasia because there may be changes or anything that grows after the 24-month period, and 24 months may not be sufficient enough. And even though Parkinson's disease isn't as common in females, we should still test this on female models to prove the absolute validity of this experiment. And here are our citations, and yes.